Hello, everybody. Thank you so much for joining us tonight. My name is Devin Rhodes, and I'm the Director of Alumni Relations here at Susquehanna University. Thank you so much for joining us for the promise of immuno-oncology. Can it deliver the cure with Ahmed Alkhateeb? Also from Susquehanna University, we have President Jonathan Green and our First Lady, Ms. Lynn Buck. We also have Jan Reichert-Brown, Melissa Kimura, Susan Kreischer, Jody Swartz, Josie Fertig, Leslie Imhoff, and Chris Markle. So thank you to my colleagues for joining us as well. And before I turn over the program, I just want to let you know that we will use the Q&A and also the chat functions to ask questions at the end of Ahmed's presentation. So now it is my pleasure to introduce our moderator for this evening, Jacob Trevino, who is from the class of 2002. At Susquehanna University, Jacob majored in physics and mathematics and minored in art history. Jacob is currently the nanofabrication facility director at New York University, a principal scientist at Palleon, an adjunct associate professor at Columbia University in the Department of Electrical Engineering, and he is the founder of the Nanotech Alliance. Jacob is also a member of our alumni board. After you, Jacob.
Thank you, Devin. Really appreciate the opportunity to be here tonight. Looking forward to Ahmed's talk. It's a great honor for me to be able to introduce a fellow Susquehanna alum scientist. And so I'm just going to give you a little background on his path to where he is now. So Dr. Ahmed Alkhateeb is a class of 2007 graduate of Susquehanna. While at SU, he majored in biology and minored in biochemistry and healthcare studies. Ahmed earned his doctorate from the Pennsylvania State University College of Medicine in molecular biology. He then went on to do his postdoctoral training at the Harvard School of Public Health and Harvard Medical School. His research has focused on immuno-oncology, the topic tonight, but also iron biology and in vitro model development. For his postdoc, Ahmed joined, or following his postdoc, actually, he joined a pharmaceutical company called Palleon. It's an early stage immuno-oncology biotech, and he served there as Associate Director of Business Development. Currently, he serves as the Director of Oncology External Innovation at Sanofi, where he's responsible for the search and evaluation of emerging technologies to treat and potentially, hopefully, cure cancer. So with that, I'm going to hand it over to Ahmed and go for it.
Thanks, Jacob and Devin for organizing this. It's always good to be back at SU, even virtually. So it's really appreciating this. And based on the list of names that Devin shared with me, people who registered, I noticed a lot of people that I went to school with, and it's really good to reconnect with everyone, and I hope everyone is staying safe. So maybe I can share my screen really quick, and let me know if you can see my slides. Yep. Okay. So for today, maybe before I also begin, maybe just add to the introduction to Jacob's words. So I did my PhD in immuno-oncology back in 2010. So that was back when the concept was still emerging. So I kind of stumbled on a project, and that got me interested in the space and start following it through. And after I finished with academia and moved to the private sector, I worked at an immuno-oncology company. So we were focused on making therapeutics based on immuno-oncology and invented some patents in that space. And part of my job right now is to stay on the forefront of where things are. So I thought that with my perspective, I can provide maybe an update on where things stand with immuno-oncology. I think it's an area that's exciting. People probably start hearing about it in the news. I'm sure that it's going to continue being in the news in the future. So giving people maybe a primer on the topic would be very helpful. But for today, I'm going to keep things high level. I'm going to focus on a few concepts that I think are important. And so you're not going to see a lot of hardcore molecular biology in this. And what I hope people would take out of it is an understanding of what immuno-oncology is and why it's important. So in the title, I actually had a debate with myself about whether or not to use the word cure, because the word cure is problematic for us as scientists. We're trained to be skeptical and empirical. So, the word cure is a little sensationalist in its nature. You see it, you've seen it in headlines, and most of the time they were not cures. So, we try to avoid using the word, but I hope that people will understand why I'm even using it in the title. I do believe that immuno-oncology has the potential of being curative, and I'm going to provide people with the actual data that support this idea. So maybe to begin with, and just to give a quick history on the topic, the Nobel Prize in Medicine in 2018 was given to James Allison and Tasuku Honjo for the discovery of something called checkpoint inhibitors, which is a class of drugs that would be considered an immuno-oncology drugs, but it's the first form of immuno-oncology drugs. And usually the Nobel Prize is not given to people who discovered drugs. Usually the Nobel Prize does not calculate how many people have you saved, and the prize is given based on that. Those two scientists also had a lot of relationships with the private sector. The Nobel Prize usually stays away from those type of situations. They usually look for breakthroughs in biology that would enable research in an entire field. So for them, for Allison and Honjo to get the Nobel Prize, a few years after the success of the drugs in the clinic, just highlights how monumental their work has been. I mean, it opens the door for us to think about cancer in a different way, and even start contemplating whether or not it's going to be cured in the near future. And that's not what they've done is something that is truly monumental. And just to put immuno-oncology in the right perspective, maybe we need to look at the history of how we've been treating cancer. So cancer, the first reports of cancer, we probably know of it from 3,000 years ago or so. And even in those reports, we know that surgery was the primary intervention. And this has been the case for thousands of years. We cut tumors out. That's what we do. And in the early 20th century, when we discovered radioactive material, we started burning tumors with radiotherapy. So at that point, it was surgery and radiation are the primary two modalities. And then in the 40s, we developed therapies. And I know that I have multiple kind of circles here. In my mind, chemotherapy and targeted therapy are kind of part of the same class of approaches. There are things that you infuse in the IV and intravenously, or you give it to patients orally, but they're systemic therapies. They have different properties, but they all do the same thing. They kill tumors. In the 40s, we discovered chemotherapies, primarily from dyes. And we also start using hormone therapies for breast and prostate cancer, and these were very effective. In the 70s, as molecular biology was evolving, we started understanding cancer dependency on certain genes or certain proteins. So we started understanding what we need to hit in a cancer in order to make it vulnerable to chemo or even kill it. And that led to the development of targeted therapeutics, mostly chemical matter, so small molecules. And in the mid-90s, we developed therapeutic antibodies, and that was a major breakthrough. So we started making antibodies in factories, and those antibodies bind to the surface of tumor cells. They usually bind to proteins that are highly expressed on tumors. And they either modulate the activity of the tumor cell, of the cancer cell itself, or they kill it. And up to 2010, that's how we treated cancer. So these are the three main pillars of cancer treatment. And if you notice that all of those things are tumor-centric. So we're hitting the tumor itself. We're cutting it, we're burning it, we're poisoning it. We're throwing everything at it. We're trying to understand how it grows. We're trying to understand what changes happen inside the tumor so that we can exploit it in the treatment. Immuno-oncology came in and added a fourth pillar, and that's why it is transformative. It's a new way of thinking about cancer therapy. Here, the drugs are not focused on the tumor itself. The drugs are focused on inducing an immune response against the tumor. And the thinking here, when it began, when Hanjo and Allison were working on this in the late 80s, it started with a simple question of, well, our immune system is very powerful. It's been evolving for 200 to 500 million years. It's fine-tuned by the Cold War that happens between us and pathogens. So it's very effective. So why do tumors exist? Why wouldn't the immune system destroy tumors before they become, gigantic? And we know that is true. The immune system is capable of detecting cells that are proliferating or dividing very fast and destroying them before they become clinically visible. And also they noticed that if you take any tumor, any large tumor, and you section it, it's actually full of immune cells. There are a lot of T cells in there, a lot of macrophages, a lot of dendritic cells. So they asked the question of why do these immune cells don't kill the tumors? It should be obvious that there should be some effect for those immune cells on cancer cells. And if you're a molecular biologist and you look at this, your first thought is that there is a signal that goes from the cancer cells to the immune system to shutting it off. So they started doing work where they were looking at the interactions of cancer cells with the immune system. And from there, they identified that there are proteins and signals that are found on the cancer cells that shut off the immune system. And that was the foundation of immuno-oncology. We started understanding that cancer cells are coating themselves with things that would make them invisible to the immune system. And so the therapies are focused on inhibiting that interaction. So the biggest type of, this is why we call them checkpoints. They're basically proteins that prevent the immune system from killing whatever cell expresses this protein. And in the earlier years, there was one checkpoint called CTLA-4, which was the first checkpoint inhibitor. It's very toxic. The second generation, which were the PD-1 inhibitors, were the ones that are the most exciting, and these are the ones that are making an impact in the clinic right now. And this is how they work. So normally, a tumor cell is mutated. It carries these mutations. It introduces a protein called an antigen that would basically, it's almost like an ID for the cell. It's an ID that this cell is not normal. There's something wrong with it. And this is what the T cells are always looking for. So the T cell sees this antigen, and then it wants to destroy the tumor cells. But the tumor cell expresses a protein called PD-L1, which binds to a receptor found on T cells called PD-1. And that produces a negative signal. And as a result, the T cell does not induce a killing program. It does not kill the cancer cell. It just basically sits there and confused about what to do. It doesn't do anything. So the drugs that we've developed, the PD-1 inhibitors, are antibodies that bind to PD-1, and there's some that bind to PD-L1 and prevent that interaction from happening. So the only signal that the T cell sees is the on signal. It doesn't see the off signal. As a result, you have tumor cell death and tumor cell killing. So this class of drugs, which are the PD-1 checkpoint inhibitors, there are 7 that have been approved in the United States starting from 2014. So these are very recent drugs. And they've been approved in over about 20 different cancer indications and cancer indications. So basically, not only we're not talking about cancer types alone, but also where they fit in the treatment paradigm of certain tumors. And they're very lucrative. I mean, they are right now bringing in about $30 billion in sales a year, about 25% of the entire oncology market. And these are drugs that were introduced in 2014. So they're growing very fast. They're expected to reach 35% of the entire market, if not more, based on current trends. So there is a possibility that they will represent most of what doctors prescribe for cancer patients. And just to give an example of the kind of clinical data that we saw with those drugs early on, the clinical development for those drugs was actually kind of interesting. It started in metastatic melanoma. So basically melanoma that spread to multiple sites. And that's where the initial trials were, and that's where the initial successes were. Partly because melanoma is one of those tumors that's highly mutated. It has a high mutational burden because it's exposed to the sun. So it collects mutations at a very fast rate. As a result, it looks very, very different to the immune system. So it looks very foreign. the immune system is triggered very quickly and is trying to destroy it. But it's a small tumor type indication. So it's not a lot of patients that get to melanoma every year. When that success happened, the companies developing those drugs, and primarily Merck and BMS, they went in and they performed trials in every known tumor type that we know of. They basically just went and ran hundreds of trials in parallel. The biggest prize in oncology is lung cancer. So lung cancer is, metastatic non-small cell lung cancer kills every year more than the US lost in the Vietnam War. I mean, this is one of the biggest killers in the world. So making a dent in the survival of patients who have metastatic lung cancer is the ultimate goal for any drug. So in 2018, this paper was published in New England Journal of Medicine, which is the study of using pembrolizumab, which is Merck's PD-1. The trade name is Keytruda, in first-line setting of metastatic non-small lung cancer, in addition to chemo. And this Kaplan-Meier curve, maybe I can walk people into like how to think about Kaplan-Meier curves. They're grotesque graphs, because what you're looking at here is 600 patients, about 400 in the combination arm. So they're getting the drug that we're testing, which is the PD-1 inhibitor plus chemo. And the placebo combination is basically chemo alone. And what you're looking at here is follow-up. So the x-axis is the duration of how long we followed the patients, and the y-axis is the percentage of the patients that are still alive. So every time you see this curve drop, it's because an event happened, death, sometimes multiple deaths in the same time. Every tick is a death here. So you're looking at basically 600 families dealing with cancer. And by the end of the study, which is almost a year and a half later, half of them are dead. So this is just to give you an idea of like what those trials look like. I mean, they're gigantic operations, and it's and for most trials fail. So to see a trial that succeed is a big deal. But back to the study. So what they've done is with metastatic non-small cell lung cancer, it's deadly. It's within the first year. Normally, half of the patients are dead in the first year. And that's what you're seeing here. So if you look at like the 12-month mark for the placebo combination, which is the standard of care, this is what patients would get normally, you're looking at 50% dead in the first year. And when you combine chemo with a PD-1 inhibitor in the first line, you're at 12 months, you're dealing with about 75% still alive. So what you're doing here is that you decrease the probability of death by 50%. This is kind of like the kind of results that you don't see that often when you're running oncology trials. I've seen drugs that were approved by the FDA where both arms started the same spot, met in the same place, but there was separation in the middle. Any improvement on survival is the FDA wants to see that. I mean, this is the reason why we develop cancer drugs, is to improve survival, even if it is for a month or 2 months here and there. So to see an effect like this was truly significant for the field. But is that enough for us to start talking about cures, or that this is transforming how cancer should be treated? It could be that it's just one good drug. Did they unlock some insight about how cancer works that maybe would enable us to start thinking about cures? Now that's a more difficult question to address, and I would say that it's important maybe to pause here and think about what the cure for cancer looks like. I think a lot of people have misconceptions about what that is, and maybe I can encourage people to maybe think about it for 5 seconds or 10 seconds. What would that drug look like? Because it has to be a drug. What would it look like? It's something that you take, and what are the characteristics of it? What outcomes should you see in a clinical trial for it to be a cure? That's not a trivial question, and I think a lot of people have misconceptions. Like, some people think that the cure for cancer is a drug that you take and prevents cancer from ever happening. That will never happen. Cancer is an evolutionary disease. it will always be there to some extent. It's caused by external stimuli that sometimes we don't know of until 30, 40 years of, of being exposed to them. There are genetic predisposition markers for cancer. And some people believe that cancer is just a disease of old age. The older we get, the more likely we are going to get cancer. So cancer is not going to go away. So what would that cure look like? And I would say that for a cure to be a cure for cancer, it has to fulfill 3 criteria. And so let's maybe start with a scenario and say that, someone is 65, they feel a pain in their back or their abdomen, they go to the doctor, they do a scan, they identify a tumor. The surgery is, you cannot do surgery because the tumor has metastasized. So surgery is not curable, radiation is not curable, and you have to take a drug. And you take a drug and it works. The first thing that it has to do for it to be a cure, it has to induce a complete response. So that's actually the technical term for when you give a patient a drug and then any sign of cancer disappears. So radiological or biochemical, which basically on a scan, the scan has to be clean. And if you're doing a biomarker in the blood, that has to be as well 0. You have to, basically the doctor has to tell the patient with confidence that they are cancer-free. So that's the first thing that you have to fulfill if you are a cure for cancer. But with that being said, there are a lot of drugs that we take, that we give to cancer patients, where you see a complete response, for 3 months, 4 months, 5 months, and then cancer comes back. So the second criteria that it has to fulfill is this complete response has to be maintained indefinitely. You have to stay cancer-free forever, basically. But and some would say, well, that's the cure for cancer, but that's not it still, because if I'm giving you a drug and it's an IV infusion every 3 weeks, you have to take this drug indefinitely. As of now, if a drug works, the patients are on it until the drug stops working or they cannot handle the side effects. So there's the idea of I'm going to take a drug and I'm going to stop it because I got cured doesn't exist. That's not something that we do. So the third point is that after this CR has to be maintained after discontinuation of therapy. So that is literally what the cure for cancer looks like. We haven't had a drug that was able to do this before. All the drugs that we have, especially for metastatic disease, again, surgery, radiation are curative. And if you take a drug in combination with surgery and radiation, there is this potential for cure. But I'm talking about stage 4 cancer where it spreads, surgery and radiation are not possible. We don't have any drugs that are truly curative. We don't have drugs that would accomplish these 3 things. If you establish a complete response and stays complete response, we usually don't take you off the drugs. So, why am I asking the question if immuno-oncology is the cure, or at least identified what the cure is because of this. So this is the longest study following up patients that took pembrolizumab, Keytruda, in metastatic melanoma, and that was, I think, the trigger for the Nobel Prize. But this was a very interesting study. What they've done is they've taken all the patients that took pembrolizumab during the clinical trials, back in 2011, 2012, and followed them for about 4 or 5 years. So out of the patients that took it, I think there was, like, in this study, there may be 600, about 600 patients. Out of the 600 patients, 15% accomplished complete responses. Some people might think 15% is small. These are really high numbers for a complete response. To give a drug and 15% of the patients that take it experience a complete response is very rare. I think in this study, the overall response rate was about 40–50%. Oncologists define response as the shrinkage of tumors. So, 40–50% of patients benefited from the drugs, but there were these 15% that accomplished complete responses. They showed up to a doctor filled with tumors in their lungs, in their livers, maybe in the bone, and all of those tumors disappeared. Again, so it checks the first box. What happened next is they continued on the drugs for about a year. On Keytruda, every 3 weeks, they're taking a PD-1 inhibitor. The complete response is held. So it checks the second box. The third box is the tricky one. So what happened is that the doctor asked them if they're interested in discontinuing, and about 90% of them said yes and discontinued the treatment. And then they followed those patients for 2 years after they stopped taking any drugs for cancer. And 90% of those were cancer-free with a complete response. So that's a practical cure. This is literally a cure for 15% of metastatic melanoma. Of course, this is only in metastatic melanoma. And I mentioned that melanoma is one of those tumors that's highly mutagenic. So it's full of mutations. The immune system is easily triggered by it. I'll show you the immune cycle later on, but you'll realize that those drugs are working on the last step in the immune cycle. So if the tumor is highly mutagenic and can induce an immune response, and then it's holding back the last step of destruction by the immune system through L1, PD-1 interactions, pembrolizumab or other PD-1 inhibitors could work very well on it. But this is the first time we were able to have data to show that there's a class of drugs that have the potential of being curative in the strictest definition of the word, a cure. Something you take, the tumors go away, you stop taking it, it doesn't come back. And that's actually very transformative. So maybe so this is PD-1, L1, and CTLA-4, and this is where we are right now. So it's we have several drugs that worked, and they are being uptaken by doctors and patients very quickly. They're generating impressive results across the board, curing some tumors. But what's next? So once that happened, there was a gold rush in biotech where there are billions of venture money that flow into biotech, especially in immuno-oncology, to search for the next PD-1 checkpoint. So in other words, can we develop another drug that if we combine it with a PD-1 inhibitor, we take the response rate, the complete response rate, for example, in melanoma, from 15% to 30, 40%. Can we cure 30, 40% instead of 15? So the quest became, what other checkpoints can we identify? And this slide, it's busy, and it's busy for a reason. And it's, the text is small also because it doesn't really matter what those names are. But these are all of the checkpoints that are currently being investigated or been investigated in the last 4 years. Every single line is an interaction between a receptor and a ligand, either found on T cells and then and tumor cells, or T cells and antigen presenting cells. And this list is growing very fast. Most of those things failed. And every failure, we're talking about $100 to $200 million in investment. Most of them failed. Out of this list, I think the only thing that generated a positive signal was a molecule called TIGIT, and it generated data in lung cancer in April of 2020. So it's a very recent development, but it just shows you how biotech works. Something worked. We went, invested tens of billions of dollars, investigated dozens of different potential mechanisms of inhibition, developed drugs, took them to the clinic, and then one after the other start failing. We're dealing with a 90% failure rate most of the time. And the goal is to go through those innovation cycles. You're basically throwing everything at it, seeing what sticks. And maybe out of that first cycle, TIGIT is the only molecule that will make it to the finish line and become a drug, and it will be combined with PD-1 eventually. And now we're back to the drawing board, thinking about new ideas and coming up with, what did we learn from the first round of failures, and can we learn from it and invest our money in a smarter way? So usually what happens is that academics identify a target, venture capitalists come in, seed a company around it, the company builds a drug, takes it maybe to the clinic or right before the clinic, then big pharma comes in and helps with the development. So everyone is sharing the risk and across the board, and hopefully in the next couple of years, there's going to be another potential success. But maybe here I can switch over to a little bit more of a conceptual view of how we think about this. So this is not just targets, but there's also a conceptual way of thinking about immuno-oncology in general. And this is something we call the cancer immune cycle. And this is how we think about the interactions between a tumor cell and the immune system. So like I said, cancer cells mutate all the time. They're dividing very fast. And as a result, they accumulate mutations. Those mutations, some of those mutations would change the shape of a protein. And every cell in our body have this system. It's almost like an ID system where it basically randomly chops different proteins that are found inside of it and then presents them on its surface as a way to say, this is the proteins that make us. And during development, early on in development, the immune system gets exposed to all the proteins that make me or Jacob, for that matter. So we're all very different. This is why we can't take immune cells from one person given to another person. It's because there is this education stage early in development where your immune system basically becomes educated to what are the antigens that are self-antigens. So cancer cells are mutating and potentially they can make a change. And this change would make a protein shape look different. And that's when it triggers the immune system. The immune system goes and says, we've never seen this before. This is new. And then it becomes a question of, is this self or is this non-self? So this is what we're seeing here in that first step. You have some release of antigens from the tumor or the presentation of antigens. There are these immune cells called antigen presenting cells, dendritic cells, which basically take up those antigens and then they put them on receptors. Then they move to the lymph nodes. The lymph nodes are filled with lymphocytes and T cells. And these are the T cells that have receptors that recognize things that are non-self. And the antigen presenting cells starts basically sampling this new protein or this new shape on those T cells to see if they can find a T cell that would be reactive. If it finds a T cell that's reactive, that T cells get activated, it starts proliferating very fast, and then it moves from the lymph node, gets into the blood, and then it's trying to seek what to destroy. They know that they identified something that's non-self, and these T cells have to go find, where this non-self is. There's inflammation usually around the tumor, produces signals that would basically traffic T cells out of the blood into the site of the tumor. So the T cells get to the tumor. And that's when PD-1 or L1 or those checkpoints work. These are proteins that these tumor cells would give to activated T cells and say, stay away from us. So for us, when we're thinking about immuno-oncology and for innovating in the space, we're thinking about it holistically. So we're thinking about ways to enhance antigen presentation. We're thinking of ways to enhance T cell infiltration into tumors. We're trying to understand whether or not cancer cells basically shut off inflammation around them to prevent those signals that tells T cells to come in and help out. We're looking at mechanisms to see if this ID system that, basically do cancer cells shut off that ID system? Basically, they say, you know what, we don't want to give an ID. We don't want to present an ID. We just want to basically exist in the dark. We don't want the immune system to know that we're here. So all of those are basically avenues for us to innovate in terms of new therapeutics and how we think about immuno-oncology. So like in my current job, I'm looking at this from every angle. I look at mechanisms that are tumor intrinsic, mechanisms dendritic cells, as well as T cells. I'm looking for new checkpoints. And it's a very, there is a lot of work done in immunology. The problem with immunology is that it's a high failure rate. It's a highly reproducible space to some extent, partly because the mouse immune system is very different than the human immune system. The human immune system is also context dependent. So if you take T cells out of the blood of a donor and put it in a dish, that T cell will be behaving differently than it would inside of a patient or inside at a tumor site. So there's a lot of context dependent behaviors with immunology, with immune cells, which makes studying them in a lab very, very difficult. This is why doing clinical trials are the most informative form of experiments for us, because that's, we're seeing how the immune system works in a cancer patient at a site of, at a tumor site. And maybe we can spend a couple minutes also discussing what's beyond checkpoints. So checkpoints are the primary kind of class of drugs, and these are antibodies, most of them, and their goal is to remove the brakes off of the immune system at different steps. But there are other ideas and technologies that were developed in parallel to checkpoints. For example, adaptive cell therapy. Probably people heard about some cancer therapies that involve taking out T cells out of a patient, genetically engineering them, and then infusing them back. These are therapies that were very effective in hematological malignancies, blood cancers, very expensive, partly because, you know, you need to take cells out of a patient, do genetic engineering, what in a closed setting, so and infuse those cells back into the same patient in less than a month. So it's a logistical nightmare. Manufacturing is a major issue. Some companies have been evolving. You know, after the first wave of companies got acquired, there are new companies that are trying to come up with off-the-shelf adaptive cell therapy. So basically, T cells they take from donors, engineer them to become, you know, so that you can fuse them into any person. But the problem there is, like I mentioned, T cells they really don't like it when you put them in a new person. Like, they will start attacking things left and right. Another modality is cancer vaccines, and this is not to prevent cancer from happening. This is more of a therapeutic intervention. So as I mentioned, if you know, if the beginning step in that immune cycle is the presentation of an antigen, so the view was, what if we present that antigen ourselves? So we know that there's a protein that's overexpressed in a tumor type. We can go and make vaccine, give it to a patient. The patient will induce an immune response against this protein, and then the whole cycle will start. If we combine it with PD-1 inhibitors, you'll have an effect. And finally, there's, you know, some work on oncolytic viruses, and this is work that's been going on for a while. Has not produced good data yet, but, you know, we've been engineering, you know, we've been using viruses as tools for a long time. But, you know, these are domesticated viruses. These are viruses that don't really infect humans or replicate even in humans, but we use them as a delivery mechanism for whatever genetic material they have, and we engineer that genetic material to be anti-cancerous. So we use viruses to kill cancer, and then we use the fact that the immune system, you know, gets reactive towards the virus itself, and then that induces an immune cycle on its own. So you know, so the space is evolving. It's moving in a lot of different directions, but what's hopeful here is the fact that, you know, with biotech, there are these cycles of innovation that happens every 3 to 5 years, where, you know, we come up with ideas, we invest in them, we try them, most of them fail, and then we learn from those failures and repeat. And I would argue that if we have the ability to do this for another 20 or 30 years, we will go through several cycles of innovation, and eventually we're going to get to several drugs, 3 or 4, that will, you know, get cancer under control. And you know, the beauty of this is right now it's actually almost the golden age of biotech. So not only you know, immuno-oncology was a huge, you know, accomplishment and got a lot of attention to the space, but also even with COVID, I mean, we're recognizing now that investment in biotech pays off in ways sometimes that we cannot even imagine. You know, some of for example, BioNTech, which is the first vaccine that was developed against COVID, it's a company that is making cancer vaccines. I mean, this is why they're in the vaccine business. It's a cancer company. They made mRNA vaccines against cancer, and it was easy to make mRNA vaccines against COVID. So I think the space is just going to keep getting more and more investment with time, and hopefully we'll get to those 20, 30 years of continuous innovation. And finally, I just maybe like as the last slide, I wanted to kind of give an idea of where things are headed in general in terms of where cancer is. You know, will we be dealing with cancer all the time? And I would say the 2 major advances here are you know, these 2 things. Early detection. I think early detection, we know that surgery and radiation, especially for early stage cancer, is curative. So early detection is the best way to fight cancer. And people have been talking about this for the longest time. We know that mammograms save lives, for example. There are 3 companies that actually started in the last 3 years that were built on top of high technology, driven primarily by genomic sequencing. This is Grail, Thrive, and Phenome. And what they're doing is they're trying to find signatures in the blood that would detect cancer before it's ever detected on a scan, a radiological scan, or even before it becomes a palpable tumor. And these three companies, I mean, they got billions of dollars in investment. They're running gigantic clinical trials with tens of thousands of people, old people basically, and they're following them for 10 years to see if they can predict who gets cancer, who doesn't. But the initial data look very promising. I mean, they're not only are they able to identify patients who have cancer without any sign of cancer, but they can actually pinpoint what the site of cancer is most likely to be, because every cancer that if it, let's say a prostate cancer will start from a prostate cell, so there's a certain genomic signature that we can identify for a potential prostate tumor. So this is kind of an exciting development, because in the next 5 to 10 years, those companies will be potentially ready to work, and then they would be selling their services not to consumers but to insurance companies. That's where the model of their business model is. The real challenge with early detection is the specificity. So you don't want to be telling someone who doesn't have cancer that they have cancer. So that's where their biggest challenge is. And then let's say early detection is not possible, or it fails, surgery or radiation, and then we're going to have all of those immuno-oncology modalities that have been developed over decades to deal with metastatic disease. So I think that's where cancer is going to be in probably 20 years. So it's going to be one of those things where we're going to start seeing more early detection, and as a result, more curative intervention from surgery and radiation early on. And if it is caught late, we're going to have a set of drugs that will be able to address metastatic disease efficiently. So I think that's all I have, and maybe I'll just stop here, and maybe if there are any questions, I'm more than happy to take them.
Great. Thank you so much, Ahmed. That was fantastic. I definitely learned a lot, and I'm actually quite optimistic, based on some of the jumps that you show clinically are like you said, like they're enormous, right? That's a huge effect that some of these techniques are having in these trials. So that's wonderful. There was one question that was asked during the talk, so I'll go ahead and pose you this question. So this is from Margaret. The question is, what progress has there been on understanding why some people just don't respond while other people are cured? So is there an underlying difference in the cancer or in the immune system?
Yeah. So with the that's biomarkers development is key, because if you can zoom in on those subsets of patients, your success rates will go up. So for example, with the lung cancer study that I showed, one biomarker that you would expect is the levels of PD-L1. So I can take a biopsy from a patient and stain for the presence of this protein. So if a tumor lights up, it's a lot of PD-L1. If it has small amounts, it has a little bit. If it doesn't light up at all, it means that it doesn't express PD-L1. So you would expect that tumors or patients that have tumors that are PD-L1 high would respond to anti-PD-1 therapies, because we know that then that mechanism is active in those patients, and that's the case, yes. So if you look at those two curves, they did this other analysis where they broke them down on PD-L1 levels, and you see a correlation. So if you're PD-1 low, you are, you're in the group of the least responders in that study. In terms of other markers, I mean, this is one of those things where if you identify a marker for why patients are not responding to PD-1 inhibitors, that's probably a signal that whatever this marker is another checkpoint or another molecule that should be targeted. So every pharma company is basically collecting, if they're running a trial, they're collecting samples from patients to try to see if they can correlate any protein level with response or non-response, because if you can identify protein that correlates with non-responders, it's a potential target for therapy. So the previous company that I was in, that's what we're doing. We're identified a certain signal in the non-responders, and we're going after it. So in terms of our understanding at the moment, not really. A lot of things failed. Like even the PD-L1 status is a nice indicator, but it doesn't work all the time. Not in every tumor type. There was another biomarker, which is tumor mutational burden, which was kind of an interesting concept. What they've done is they would take a biopsy from a tumor, and then they would look at the number of mutations that the tumor accumulated. So the idea is the more mutated the tumor is, the more likely it is to induce an immune response and be responsive to PD-1 inhibitors. Like I mentioned, melanoma has the highest tumor mutational burden of all tumor types. So that was an idea that was, people played around with it in 2019. Looked interesting someday, but it was not conclusive. Aside from that, I can't really think of anything else that was kind of a serious predictor of response.
Gotcha. Great. Okay. So a couple new questions just came in and comments. So I would ask you to read them later because there's a personal one for you, thanking you for giving this wonderful talk tonight from the president and his wife. And yeah, you could take a look at that afterwards. There's a question from Pete McQuaid, and he wants to know, can you discuss immuno-oncology specifically for treating pediatric brain tumors? And he asked this because he's actually president of a board of trustees for the Courtney Rose Foundation, and which works in this space. And so there's a longer part to this question, a comment actually that you should just check out afterwards. But yeah, I mean, is there anything specifically that you know about for treating pediatric brain tumors?
I know that with PD-1, the gliomas been very difficult. CAR-Ts, I know that there's some work in CAR-Ts. So these are those adaptive T-cell therapies where you take a T-cells, engineer it, and to attack the tumors. I've seen some, yeah, I think CAR-Ts are the primary kind of focus with brain tumors. Brain tumors are tricky for a lot of different reasons. I mean, it's also an immune privileged site. You don't have a lot of immune cells inside the brain, so getting T cells there is tricky. But yeah, I don't know much about glioma, to be honest. So
okay. There's a question from Julie about asking what your thoughts are on genetic testing for cancer. She's hesitant to be tested for markers because currently there's not better prospects than there was 20 years ago. Right? We've made progress for sure. But do you want to know? I mean, this is a good question.
Well, I mean, like the BRCA1, 2, that's a very well established marker. And not only this, I mean, we have therapies that work on BRCA1, 2, tumors. So, I mean, it depends. I mean, it depends on what kind of testing you're doing. There are a lot of association studies out there, and maybe people have to recognize that like not every association is going to pan out, and sometimes they're underpowered. So there are some markers for cancer that we know are very strong. So I don't know what type of genetic testing she's referring to, but I would focus on established markers. And the susceptibility it's a very complex thing. So just genes alone are not it. You're talking about environment, diet, age. So there's a lot of different factors. But there's some things that are very, highly, there are some very strong genetic indicators, and that, if you're worried, if you have any history in the disease, it's probably a good idea to get them checked. But, and with that being said, I'm not a clinician or a genetic counselor, so I think take my advice with a grain of salt.
Great. There's a question from Daniel Decker. Wants to know: is there a negative effect to using too many therapies together? So the first reaction would be, let's use as many as possible to bring that number down. But is there a point where they start to work against each other?
Yeah. I mean, toxicity is the limiting factor. So when you're doing combinational studies, there are certain limits for tox that you need. So if you want to, so the best drugs to combine are the ones that are not toxic. So non-toxic drugs, you can go to 3, 4. Like I give you an example of multiple myeloma, for example. I mean, the first line of treatment there sometimes can go to a quadruplet. Like you can hit a patient with 4 different drugs. and then scale it down if the toxicity is too much. But tox is the main thing. With immuno-oncology agents, since the toxicity might be immune mediated, some toxicities overlap. So like having two drugs that are hitting similar mechanisms or have immune adverse events, some of those toxicity profiles might overlap. So as a result, the toxicities are not additive, if that makes sense. They're kind of like synergistic in the good way. Two are not as toxic as the addition of both together. So, but every drug is different. And with immuno-oncology, we're dealing with some of them are weird side effects, but for the most part, they're relatively safe. You're dealing with rashes and inflammation in the gut, but they're relatively manageable compared to other drugs that are used in cancer treatment.
Thank you. This is just a comment from Jan Reichert Brown. She's done very well. She's thrilled to see the scientist that you've become. It's a very nice comment. Thank you, Jan.
So Jan was actually my advisor and my senior advisor. So I'm glad to. I'm happy to hear this. Yeah.
That's great. Let's see. I don't believe there's any more questions in the Q&A right now. I have one question. So the PD-L1, and I'm not a biologist by any means, but it strikes me as sort of like this prime target for finding a tumor, right? So it reminds me sort of the spike protein on the coronavirus, for example. Are there ways to bind to that and then have that binding agent be sort of another binder for another drug to come in and do damage?
Yeah, like the issue with PD-L1, it's kind of this is a system that exists independent of cancer. So anytime you have inflammation, and whenever you have T cell activation, PD-L1 goes up in that entire area. So it's basically an off signal for the immune system. So it's part of a normal mechanism. It's just cancer figured it out. They basically figured out that if I just keep making this thing, T cells keep me alone. And it's kind of an evolutionary process where they just figured out this simple trick that would keep T cells away. And what we found out that this simple trick, its impact is enormous. And tumors are not, again, like they're not thinking or they're not evolving. They're just following evolutionary principles. But they didn't really factor into the fact that they're dealing with an organism that evolved how to think and how to manipulate environment and create antibodies and understand what's happening and hit it. So but it is part of a normal mechanism. Yeah. So targeting it outside of cancer might be tricky, or using targeting it to deliver toxicities. You're going to have toxicities all over the body in a way.
Yeah. Gotcha. Let's see. Don't see any other questions. Okay. Well, if there's no other questions, oh wait, let's see. We have a comment that came in from Kristen. Says, thank you very much for explaining a very complex subject in layman's terms for us to better understand. She graduated with Pete McQuaid in SU in 1985. The Courtney Rose Foundation is the foundation in memory of their 9-year-old daughter. So thank you. Yeah. So if there's no other questions, I believe, well, actually, let me say, is there a way that people get in touch with you? What's the best way if people have questions afterwards, or they want to stay on top of what you're doing? Is there a way to stay in touch?
Yeah, I mean, social media is a good place to be. Yeah, social media I think is reasonable.
Okay. Do you use Twitter or?
Twitter and Facebook, I'd say, yeah.
Got it. Okay, gotcha.
And
okay.
What about LinkedIn? So I know we have some current students, and can they go ahead and reach out to you on LinkedIn?
Of course, and if there's students who are interested in thinking about doing a PhD in biomedicine, happy to to talk and share my experience for sure. So.
well, we'd like to thank Ahmed one more time. So I'll give him a round of applause, virtual round of applause. And we'd like to mention that, well, thank you for attending this talk. And this is a series of talks. So we're really excited about the upcoming lineup. And if you registered for this, I'm sure you'll get follow-up emails to let you know that those are coming through. I myself will be giving a talk in a few months, and yeah, I'll hand it over to Devin if he has anything else to add.
Yeah, no, thank you both. Thank you, Jacob, for moderating. Excellent job, Ahmed, as always, just incredible. And I echo what everybody said. You explained everything so that anybody can understand, and that's very much appreciated by everybody. So thank you. And no, both of you, great. Thank you. Thank you, everybody who attended tonight. I appreciate you taking the time. And yeah, stay tuned for more incredible webinars from our amazing alumni. So thank you.
Thanks a lot.
We'll see ya. Have a great night, everybody.